All right, so welcome to McMaster Engineering. I guess you've had a long day, so this is the last one. My name is Dr. Todd Hoare. I'm an assistant professor in the Department of Chemical Engineering. I'm also cross-appointed to the School of Biomedical Engineering. So those of you who are interested in biomedical engineering, biomaterials, I'm going to be talking a little bit about drug delivery today. But we do a range of things in biomedical engineering, ranging from imaging, so sort of the MRI kind of stuff that you might have seen in hospitals, all the way down to biosensors, prosthetic limbs, drug delivery, growing new tissues using materials. So it's a range of activities that we do. But my home department is chemical engineering, so I'll actually be talking a little bit more about what chemical engineers do and how biomedicine, which seems to have nothing to do with chemical engineering at all, if any of you, sort of at least the prototypical, the traditional chemical engineering, it's a little bit different. But I'll show you how that kind of fits in. Okay, so engineering in general is essentially the use of the scientific discoveries and practical knowledge to create things like devices, structures, processes, materials, and systems, right? And the key idea behind engineering is that we should be benefiting people with whatever we do, right? So we're always trying to make a product that makes somebody's life better in some way. Whether that's driving across a bridge and not having it fall down, which is always handy, or for what I do, essentially how to formulate drugs in different formulations so that instead of injecting 3 times a day, you have to do it once a week. Instead of having drug go everywhere in the body, so like a chemotherapy drug, which is very hazardous to normal cells, instead it goes right to the cell that it needs to go to and kills only that cancer cell. So that's how we try to make, have benefit to people. This is generally what engineering aims to do. So chemical engineering is essentially trying to take advances in knowledge in chemistry and apply that to make useful products. And when I say the word chemistry, half of you are probably saying, oh, chemistry, no, make it stop, get me home. That's the general response I get. When I tell people I'm a chemical engineer, they're like, oh, I'm sorry, you have to do chemistry. And to some extent, what I do, it does have a decent amount of chemistry in it. But broader, chemical engineering is about processes and large-scale processing systems, okay? So some people in chemical engineering never touch a single chemical in their whole lives. They spend their time in front of a computer trying to figure out how to use mathematical models to make processes, often chemical processes, but there's also other kinds of processes run more smoothly. Okay? So I like to call chemical engineering more process engineering. We tend to look at sort of big processes more over than not. And it's probably one of the most diverse engineering disciplines. There's all sorts of things. So the chemical process is certainly the traditional stuff. So if you know anyone who's a chemical engineer, particularly the older chemical engineers, if your father or grandfather was, the probability was they were into oil or they're into processing materials in some way. So that's sort of the conventional chemical engineering. And we still do that. There's a lot of new stuff that chemical engineers do, things like physical processes. So what you see here is actually a gigantic membrane water filtration unit. So Hamilton is about to spend $700 million installing things very similar to this in our wastewater treatment plant to take the water from Hamilton Harbor, Lake Ontario, and bring it in so it's safe for us to drink. This is a large-scale process, but it doesn't really have much to do with chemistry. It's about designing the membrane, designing the pore size, controlling pressures, controlling the whole process to make it happen. Biological processes. So this can be things like soil remediation, so if there's some sort of contaminant in the soil. Also things like wastewater treatment as well, or the water byproducts from plants. This is actually a holding pond where bacteria, algae are breaking down toxic chemicals. Understanding how to grow those bugs, how to regulate them, how to make sure we get all the chemicals out, that's really what is really important in order to design and understand that process. And also just to control processes in general. So for example, if I need a particular flow rate out, I have all these flow rates going in, how do I control the valves? How do I tune the different parameters of the system to make this operate the best possible way? And because we're engineers, we always sort of have money in the back of our mind too, so how do we make our company the most money possible? So all these are aspects of chemical engineering. So I said this is the biomedicine or the bioengineering talk, so you're thinking, what's this guy doing talking about oil? Well, chemical engineers understand whole processes, and there's no better example of a factory or a plant than your body. So if you sort of break down your organs, look at what all these things do. So the heart is essentially pumping the blood around, so that's causing the flow in our system. The blood vessels are the pipes. They're carrying blood, they're carrying nutrients, everything that we need, proteins, throughout to nourish the tissues. The liver is essentially a chemical reactor. It's producing all sorts of chemicals. It's metabolizing things. It's breaking down toxins. It's also involved in waste disposal. The kidney is basically does the same thing as that giant wastewater treatment filter I showed you on the last slide. It's processing wastes in our blood, getting rid of them, basically cleaning the blood in time. The skin is basically our shielding wall, right? So it's keeping harmful stuff out, keeping heat in. The brain is the computer, if you will. It's the control center. Regulates all the other processes, balances them properly. The muscles are the moving parts. So those are our things that are moving up and down, that we're rotating, so on and so forth. The bone is the structural support, so the steel girders that hold our plant up. Eyes and ears are our sensors. These are all things that chemical engineers traditionally know a lot about, from like the oil industry or the steel processing industry. We can apply these same principles to biology very easily, and that's where a lot of the really exciting stuff is happening. Okay, so bioengineering in general is the application of the principles of biology to develop materials, products, and devices to benefit society and specifically your health. So this is obviously medical. This is where a lot of this stuff comes in. Anyone have any idea what this is? Yep. It is a heart valve. It looks like a ping pong ball in a cage. But essentially your heart, what you need to do, this is actually a valve that would go in between the atrium and the ventricle in your heart. So between sort of the filling and the ventricle actually pumps it out. So what you need to make sure of is that the flow can go one way only, right? You want to make sure it goes from the atria, the storage, into the ventricle. And then when the ventricle contracts to push the blood throughout your body, you got to make sure it doesn't go backwards. So look what this does. So if I have pressure coming this way, the ball goes up, it reaches the top of the cage, I get flow out. If I have pressure going this way, so the ventricle is contracting, it's pushing the ball against the support, nothing's going to go out there. So we essentially made a one-way valve that mimics the heart. And essentially this is kind of a woven support that you can suture in, and that's how they replace things like mitral valves, if you know anyone who has had a mitral valve transplant. Probably looks something similar to that. Also be things like pharmaceuticals, so there's a lot of it. Oh, sorry, is there a question?
Do you know if they're still trying to minimize like red blood cell loss due to like crushing of it?
That's a really great question. So this valve design is actually horrible for red blood cell loss because there's a lot of shear. So actually there are other designs that have been made now that are kind of slats on pivots. So they open up parallel to the blood flow, so you don't get as much shear and you save a lot of red blood cells. The problem with the other designs is they're not quite as good as this design, keeping blood flow from going the other way. So there's a trade-off. Is it more important to keep the shear low? Is it more important to keep the one way? And depending on the patient, it might be a different answer. Okay? Any other questions? Feel free to interrupt me. My undergrad classes interrupt me all the time, so no problem. Environmental processes, also fuels, so biofuels. You've probably heard a lot about people are now being paid to grow corn, and instead of having that corn eaten, or instead of that corn feeding livestock, they're fermenting that to create ethanol. And most of the time when you go to the pump now, about 10% of what you're pumping into your car is ethanol. And some of that is from biosources. So bioengineering encompasses all these things. So most of what I do is on the medical side. That's what I'm going to talk about. So bioengineering, the goals really are to understand the whole process. And as chemical engineers, we're used to doing this. And understand how living organisms interact and respond to their environments. We also need to understand how biological functions and our engineered products or engineered processes work together. So in order to be a good bioengineer, you have to know something about biology. And I'll be perfectly honest with you, I did chemical engineering as an undergrad. I knew nothing about biology. And over time, I kind of learned more and more as I needed to for what I was actually doing research in. So these are things that you can learn, but we also have the chemical engineering and bioengineering program here at McMaster, which will let you actually get a good basis in biology at the same time you're doing a chemical engineering degree. So that's a really great program to actually do exactly this. Understand sort of the relations between the engineering stuff and the biology stuff. So ultimately what we're trying to do is make engineered products. So to improve healthcare, clean energy, excess CO2, obviously concerns with that, clean up the environment, even things like bioterrorism. How to design masks, for instance, to, you might remember maybe 5 or 6 years ago, people were mailing around anthrax in the United States. So how to design masks, even paper, that can minimize the risks associated with that. Those are all bioengineering problems. So what I do in my research program, and I also teach a course on this in 4th year, it's called drug delivery. So what drug delivery is essentially controlling the rate and location of drug release in the body. And we do that by designing materials. And this can be a wide variety of materials. So implantable materials. The example I share here is the gliadel wafer. It's basically the size of a dime. I should have put a picture up. But it's probably the first controlled release thing that became commercialized. And what it was used to do is to treat brain cancer. So the doctor would go in, they'd cut out the tumor as best they could. But you're never going to get every cell when you do a biopsy, right? You're going to miss some. So what was done is these dime-like discs were put in, and over a period of the next 5 years or so, slowly, drug would release out of those and kill any cancer cells that were stuck in the brain. So if the cancer came back, if there were extra cells that the doctor couldn't exhume, they would be treated in that way without you having to have another brain surgery, which is obviously very, very invasive, very problematic. Yep?
Did that also kill the brain cells, or was it just only solely the cancer cells?
That's a really good question. At the rate at which the drug was delivered, and cancer drugs are, their main job is to kill, right? They kill cancer cells more effectively than they kill normal cells. So I would certainly not say that it didn't kill any normal cells. It certainly did. But it would kill cancer cells preferentially. And if you get the rate right, you can do a pretty good job of it. Injectable systems. So if any of you are diabetic, you're quite familiar with injectable systems, right? The injectable insulin. And some of those insulin, there's short-release insulin and there's long-release insulin, if you know anyone who has diabetes. Long release is basically a combination of insulin with a material to control how quickly it's released in the bloodstream. That's exactly drug delivery. Ingestible systems, so I'm sure some of you might have taken one this morning, right? Those Tylenol gel caps, tablets, all that type of stuff. Inhalable systems, if you have asthma, you're very familiar with these types of systems. Those are the puffers. And also transdermal systems. So the classic example of that's the nicotine patch, right? You stick it on your skin, delivers nicotine slowly over time. So why would we bother working on this? Well, there are three main reasons. One, if we can deliver a drug over an extended period of time, I can reduce the amount of times you have to take the drug. This makes it much easier for you. There's a higher probability you'll actually obey the doctor. You guys are really, well, not just you, everybody is problematic. They don't listen to doctors very much. So if they tell you, take it every 12 hours, they're like, 13 hours, I'll skip today. And this is a big problem, particularly with antibiotics, with antibiotic resistance, okay? So if we can reduce the treatment intervals, it's more likely you'll take the drug, and the drug will be more effective. We can increase drug efficacy in that if we deliver the drug to the right place and at the right rate, we can use much less drug to do a much better job, as we're targeting the specific site with the exact dose that's needed. We can also, in the same way, reduce side effects. So instead of taking one Tylenol, for instance, where maybe 1% of the drug ends up where you actually need it, the other 99% goes who knows where, right? Other places in the body. If we can target it to that place and release it at a particular rate at which it's used at that location, we can minimize the side effects in the rest of the body. So controlling location is the first thing. So we can either implant something and have the drug deliver right there. So the example of this is a drug-eluting stent. So if you've ever known anyone in your family or a friend who's had bypass surgery or they've had a cholesterol blockage, one of two things happened. Either the doctor took a vessel from another place in the body and grafted it around the vessel that was blocked. So basically, in chemical engineering terms, they took the tube that didn't work anymore and installed a new pipe to get around it. Or the other thing they could do is put a stent inside that blood vessel. What a stent does is basically like a very, very small slinky. So when you put it inside, is that still a topical reference? I'm dating myself, aren't I? So you stick it inside a blood vessel, and what a stent does, once you have it inside, it expands. Okay? So the doctor can insert it, it expands, and as it expands, it pushes the vessel out. So it doesn't actually get rid of your cholesterol. It creates a blood vessel that has a big bump on the outside. But it opens the channel so the blood can flow, so you don't have risks of heart attacks. Okay? So what we can do, yep?
Do they leave it in there?
Yeah, usually they do. So there are degradable stents which will go away by themselves. So most stents, if you put a stent in, as long as you can minimize blood clotting at that stent, you could have it for 20, 30 years. Yeah. Yep?
Is that similar to like angioplasty?
Yeah, exactly. It's the same thing. So this is actually how they do it. So angioplasty is the process by which they install stents, basically. So they put the balloon inside, they blow the balloon up, that expands the slinky. That's how they fill the space.
If it degrades over time, how would that solve the problem?
What can happen is
it doesn't get rid of the cholesterol.
That's true, but your body is not a static thing. So just to give you an example, your liver, I believe it is, every 300 days, your liver is basically entirely new. So there's an enormous amount of turnover of cells in your body. So the same thing can happen in your blood vessels. So if your blood vessel is pushed open, it can actually essentially reform the epithelium and reform the cell structures to compensate for and sort of stabilize that pushed open position. It's unusual to have something that's completely degradable. Usually something will stay in there for a long period of time, but it has been demonstrated. Any other questions? So what we can do is we got this metal stent in there, it's opening the blood flow. What if we coat it with a polymer and a drug? That's going to deliver that drug slowly over time. So if I'm concerned about blood clotting there, if I'm concerned about inflammation because of the surgery, I can deliver a drug which will address those problems very easily. The other neat thing we can do is actually what I call search and destroy nanoparticles. So if I make something really small, we'll talk about what nanoparticles are in a second, what they'll do is they'll circulate for a long period of time. So if I put chemicals on the surface of those that recognize specific chemicals on the surface of cells, they'll stick to those cells and deliver right at that site. So people have used this for chemotherapy. Cancer cells express different types of proteins on their surface. So we find stuff that sticks to those proteins, put it on, our drug delivery vehicle will stick there and will deliver drug at that place. So the controlling rate, generally what we want to do is aim for what's called zero. Yep?
The nanoparticles, how do you get around the antibodies fighting them?
Good question. So one of the central truths of bioengineering is you cannot fight the body. So stuff always sticks to anything. So all you can really do is circulate them long enough that they'll have several chances to go to the place where they need to. So just to give you an idea, if you inject a nanoparticle, a good circulation time, if you did a fantastic job, state-of-the-art technology, it'll circulate for about 12 hours before it's either taken up by the lymphatic system or goes inside another cell. That's like the best case scenario. So you're basically just letting it circulate a long time, increasing your probability that it'll stick at the site where it wants to. But you're right, eventually you will lose. The body wins. Okay, any other questions? Good, lots of good questions. So generally what we're trying to do for drug delivery is to try to get what we call zero order release. So constant release is a function of time. Why we want to do that, if you're popping pills, the drug concentration in your blood looks like this green line. So it'll go way up. There's a chance of overdosing, so it's unsafe. That's when you run into real problems. But it goes down really fastly. It's usually an exponential decay. So what this means then is only for this very, very small time is the drug concentration in the clinical window where it's safe, so it's not too high, but it's high enough that it's actually effective. So we want to try to keep the drug concentration in the blood in that safe clinical window for as long as possible. That's generally the idea. The other thing we can do is use a stimulus. That's something that we can trigger either inside or outside the body to say, hey, I want drug release now. Or, in other cases, I want more drug release now, less drug release now. I can talk to these things inside the body in order to do this. I'll show you an example. So the key challenge to control rate, we either want constant release, sustained release, or for other things, pulsatile release. So I get a bump out, it stops, a bump out, it stops. Anyone think of something that would be useful for that? Yep.
I'm just thinking about that. Say, I guess maybe if you're just saying like one part of the heart, maybe.
Possible, yeah. Yep.
Wouldn't that be like for like you said about the brain cancer with the glial? Because you only want to do it if you like they're having brain cancer.
Definitely. Yeah, the chemotherapy is hard to predict. So if you can do that for chemotherapy, you minimize side effects. Any other ideas? Insulin's another great example. Because when you're eating, you need a lot of insulin. When you're not eating, you don't need a lot of insulin. So being able to do this would really help with diabetes treatment as well. So how we do this, we combine drugs with polymers. So polymers are long chain molecules, many repeat units. And that's actually what polymer means. It means many units. Does anyone have any examples of polymers that you might have seen? Maybe you've seen one today before, yep. Silly putty, yep, that's a great example of a polymer. So an elastomer.
Any type of plastic.
Yeah, any type of plastic and polymer are often conventionally used the same way. So plastic bags, glues, paint is actually a great example of a polymer. They're polymer particles. So what we do in our lab is try to work on polymers that can be dissolved in water and use those polymers for drug delivery applications. So if you take a polymer that's dissolved in water and you cross-link it, so you take individual chains and you link them together to make a network, you create what's called a hydrogel. You can link these hydrogels together with chemical bonds, or you can link them together with physical bonds. So these are things like charges, like hydrogen bonding, which you might have talked about in chemistry. Anyone have any ideas if you've seen a hydrogel or used a hydrogel today? Yep. Yeah, anyone wearing soft contact lenses? Yeah, you're wearing 2 hydrogels in your eyes right now. That's probably the best example. Any other ideas where you might have seen a hydrogel? Anyone changed a baby recently? The lining in a diaper is a perfect example of a hydrogel. So hydrogels can take up a bunch of water. So they line diapers with hydrogel polymers that are dry. So the baby does its business. It absorbs that liquid and keeps the baby from feeling the dampness on so you don't have to change the diaper as much. So when you're parents, you'll really appreciate that. Jello is another example of a hydrogel that you might see. So just to give you an idea, if you have contact lenses, you'll know that you have to take eye drops quite a lot sometimes if you have dry eye. Or if you have to treat an eye condition, you have to take a lot of eye drops. This can be even like every 15 minutes. So when you put an eye drop in your eye, you blink once. And you blink about, on average, once every 15 seconds. So blink once, 95% of the drug, or the volume of that eye drop, is gone. So you only have 5% left. You blink again, 95% of that's gone. This is a really tough problem. So I want to deliver drug to the front of my eye. And if you have dry eye or if you have some sort of inflammation in your eye, this is often done. This is really hard to do. So something we've done is we've actually created a drug-eluting contact lens. So what this is a hydrogel there on either side. So this is the same hydrogel that a few of you might be wearing right now. In the center, there's a thin film. And you'll say, hey, why do I want to put something that's white in my eye? Well, you only see through a very, very small part of your eye, right? That's essentially where the pupil is. So as long as where the pupil is clear, you'll see the image perfectly. And those of you, I don't know if any of you have cosmetic contact lenses. Anyone changing their eye color? Essentially, they work the same way. There's a little film inside them which has a color. So if you're blue-eyed and for some reason you're very upset about that, you can have green eyes. Works exactly the same way. Okay? So what we did is we made this contact lens, and you actually see this is delivery of an antibiotic. And you can see we can get kind of constant release over many, many days. And because it's right up against the eye, your blinking doesn't keep taking it away, so it's a lot more effective. The other thing we can do is make hydrogels into nanoparticles. And when we do that, so the hydrogel looks the same. It's a network of polymers, but now it's really, really small. And by really, really small, I mean about 100 nanometers. So just to give you an idea what 100 nanometers is, if you pluck a hair from your head, that's about 100 microns. Okay? So 100 nanometers is if you divided that hair lengthwise 1,000 times. This is something very, very, very small. So this is nanotechnology, if you will. The neat part about if we make hydrogels really small, we get all the advantages of gels. So it still binds a lot of water. We can deliver drugs really easily. But because it's small, it responds really, really quickly to changes in its environment. It has a really big surface area. Right? So if I take a gram of stuff and I put it in a big block, it has a pretty small surface area. If I make 100 nanometer segments of that, it's got a ton of surface area. So we can change how the thing behaves quite dramatically. Uh-oh. I might have talked too long, and my water might not be hot enough for this to work. Well, I'll do it in a second. Hey, maybe, would you mind getting...
The taps are automatic, so I can
Oh.
get it so warm.
Perfect. I'm close to it now, so we'll do it. So what's really neat is if we can make hydrogels of what's called smart materials. So I talked about, when we were talking about drug delivery, if we can talk to something inside the body, say, turn drug release on, turn it off, turn it up, turn it down, that's really beneficial. We can do that with these smart materials. What a smart material is, we have some sort of initial state, so its initial size or solubility or conductivity or whatever it is. We apply a stimulus. That can be temperature, pH, or a concentration of a chemical. I'll show you a couple of quick examples of each. And we change the properties of that material in some way. So we make it shrink, we make it swell, we make it degrade, we make it insoluble, we make it soluble. And by these changes, we can control how the drug interacts with that material and how it's released. Okay? So the smart material we work with a lot is called polyanisopropyl acrylamide. This is the repeating unit. So we said polymer was many units. So this is the unit that appears over and over and over again in this long chain. This is a really neat polymer. So most things, when you heat them up, do they become more soluble or less soluble? More soluble, right? So you can dissolve a lot more sugar in water when it's boiling as opposed to not. However, this polymer does the opposite. So when you heat it up, it actually becomes less soluble. And that happens really, really quickly. So at about 31 degrees Celsius, it's happy, it's soluble, it's minding its own business. At 33 degrees Celsius, it essentially becomes an insoluble blob. And the other neat thing about this is it's totally reversible. So if I cool it back down to 31, it becomes soluble again. I heat it up, it collapses again. I cool it down. We've done this hundreds of times in my lab, basically until my grad students started yelling at me and said, make it stop. So this is completely reversible. It can happen over and over and over again. So what happens when I make a microgel out of this? Well, what happens is not very much until I hit this critical temperature. Then very quickly, I see the particle size go down. Okay, so we get a shrinking event. The surface charge goes up, which is what this means. The refractive index, so the turbidity, so if you look at it, it becomes kind of white instead of clear. That goes way up, and the water content goes way down. And again, we can turn this on, we can turn it off. So we can make it swell, we can make it shrink. We can do this any number of times. So there's two real consequences of this, okay? So the first one I already shared with you, which is size. So if I have something at low temperature, it's going to be big. When I heat it up, and I heat it up a very, very small amount, it becomes small. And I can switch this over and over and over and over again. The other neat thing about it is that I can change whether the microgels or the nanoparticles like to stay apart or whether they like to stick together. Okay? So the reason for this is because I'm basically making the polymer a lot less soluble when I heat it up. So when the surface interact with each other, when the particles collide, the particles will start to stick together. That's actually what I'm going to show you, and Kansas is back at a perfect time.
It's actually boiling water this time.
Oh,
so
excellent.
It's really hot.
So let's actually do this one. So you see, this is a microgel suspension. This microgel is about 200 nanometers in size, so about 1/500th of your hair. Okay? So you see it's kind of blue. So it's not clear, but it's translucent, I guess you would say. All right? What I'm going to do is I'm going to put it inside hot water. Wow, that is hot. You weren't kidding. Almost instantly, you can see this has become white. It's become a bunch of globs. It's essentially become a solid. So it's become, instead of a suspension, it's become kind of the solid blobs of polymer. This is really hot, so this is going to take longer than I thought. If I cool it down again, you guys can play with this after if you want. I'm going to keep talking because it's going to, it's got a ways to cool down. What's going to happen is, again, very quickly I recover this blue translucent state. I can do this over and over again. I can have the particles individual, I can have them stick together. So why is this useful? Well, cancer tumors in your body are a little hotter and a little more acidic than the rest of your tissues because they're metabolizing a lot. So if I can inject these, I can circulate them, I can tune the temperature at which they aggregate to the temperature of a cancer tumor. I can get these to aggregate and stick at that tumor site. So I can deliver drug right to that tumor. So that's something we're working on. So applications of these materials, I can modify viscosity. I can change how much light goes through it, and also change the color. So just to give you an example, this is a bit smaller microgel. So this is about 100 nanometers. This is blue. I can almost tell you what the particle size is by the color. So depending what color it is, you can say, oh, that's 100 nanometers, that's 200 nanometers, because it refracts different wavelengths of light. We can use that to filter out different amounts of light, to change the wavelength that gets through, to change the intensity that gets through. We can recover things from complex mixtures. We can even do like nano machines, right? So these are doing this all the time. So it's kind of a machine on a nanoscale. We're interested in it for drug delivery. So how to actually use this thermal transition to either localize the drug at the place we're interested in, or to change how quickly it gets out. So temperature responsive microgels, I just showed you an example of that. What we can do is we can load drug inside, and when we heat it up, one of two things happen. If the drug and the polymer don't stick together very well, when I collapse it, it basically acts as a pump, and it pumps all the drug out of the system. So I can get doses when I want them, very large doses. If the drug and the polymer like each other, they'll start to stick to one another. So when it collapses, the drug stays in the network. And now, instead of the drug trying to release through something that's like 95% water, now it's trying to release through something that's 20% water. It's a lot slower. So I can load a lot of drug when it's swollen, let it collapse in the body, and release the drug over a much longer period of time. I can also make things that respond to glucose. So we've actually done this. So this is a sort of a self-regulating diabetes treatment. So at low glucose concentrations, not a lot of glucose binds to our gel. and it doesn't swell a lot. As your glucose level goes up, more glucose binds to the specific binding groups that we put in. We generate more and more charges in the gel. The gel swells, swells, swells. So if I have insulin inside, as the gel swells, the pores get bigger and bigger and bigger and bigger. And as the pores get bigger, it's easier and easier for insulin to get out. So what this does is essentially, at low glucose levels, it's harder for insulin to get out. At high glucose levels, it's easier for insulin to get out. So I'm essentially controlling diabetes in this way. And this is reversible. So as my insulin works and the glucose level goes down, this is an equilibrium binding, so the stuff falls off, the gel starts to shrink again, the insulin release rate goes down again. So instead of having to regulate your blood sugar all the time, you could use these type of things to actually regulate it yourself. So you wouldn't have to do the same level of testing. The other thing that I want to show you just before we wrap up is magnetically responsive materials. And this is kind of cool. This is something that we published not so long ago. What these materials are, this is actually a membrane. It's a little hard to see. It's a membrane that's made of a plastic, which is porous. So just like a coffee filter, basically, almost. A plastic version of a coffee filter. We have those thermosensitive materials inside. We also have magnetic nanoparticles. And these nanoparticles are about 10 nanometers in size, so about 1/10,000th the dimension of your hair. So these are extraordinarily small. The neat property of these is when we place these in a magnetic field, and this is the same type of magnet that's on your refrigerator. The chemistry is the same thing. It's just way, way, way smaller. If we put it in a magnetic field, what happens is that the north and south poles of that material want to align with whatever the magnetic field is outside of it, right? So if I have north here and south here, the poles of my magnet, the south will be here, the north will be here, right? That's how magnets work. What if I switch the polarity of my external magnet all the time? Well, if I switch the polarity all the time, my magnetic particles are going to try to catch up. They're going to switch north, south, north, south, north, south. And in doing this, we generate a lot of heat. So what we can do then is turn on a magnetic field. My magnetic particles generate heat. That heat gets transferred to my thermosensitive materials. They shrink. They're inside the pores, so when that material shrinks, it opens the pore. Drug can get out. When I turn off the magnet, they stop generating heat. My material swells again, it fills up the pore, the drug release stops. This is exactly what we showed. So you turn on the magnet, you get a lot of drug out. You turn off the magnet, it stops. Turn on the magnet, a lot of drug comes out. And again, you can do this over and over and over and over again. There's no moving parts. It's just all material science that goes into this. Okay? So that's all I wanted to talk about today. So just in summary, a lot of people have called the 2000s the biological century. And it's really just begun. And there's a lot of really interesting work going on here at McMaster in different bioengineering fields and also within chemical engineering, manipulating biology and understanding processes in terms of engineering processes and biological processes to solve medical, environmental, security problems. And chemical engineers are playing a key role in that. So thanks for your attention. Hope you enjoyed the day. And if you have any questions, please let me know. Yep. Sorry? Yeah, come on up. This one's the one that works best.
So what's this?
So this should work too. You can try this as well. But so that's a much bigger microgel, so that's the only difference. So that's why it looks whiter. The bigger they are, the whiter it becomes. So the smaller it is, the more translucent, the more colored. It's more dramatic with the other one. It's already very translucent to start with.
So when you heat this up, it expands. Are the particles sticking to each other, or?
No, it's not expanding. What's happening is they're shrinking, but they're also becoming a lot more hydrophobic. So more water hating.
Yeah.
So they're still circulating, they're colliding, because any particle is in motion, right? So every time they collide at the high temperature, they stick. Every time they collide at the low temperature, they don't stick. So that's what you're seeing. You're seeing the collisions and the sticking. Yeah.
So it's more hydrophobic when it's higher temperature, when it's
Yeah.
hotter?
Yeah, because if I don't make a microgel, if I don't make a hydrogel, what happens is if it's just a linear chain of polymer, it actually becomes totally insoluble. Because it's a gel, there's sort of elasticity which prevents it from being completely insoluble.
When
It
it's
holds
heated
it
up,
up.
it becomes larger, so it can it's more like you're showing it was more porous, so the drug would escape.
The other way around. When you cool it down, it becomes larger. So it's the opposite of what you're normally expecting. Yeah.
So why does it change its state?
I'm sorry?
Why does it change from a liquid to solid?
Why? Well, the reason is that essentially the monomer, the building block of the polymer, it doesn't know what, it's kind of schizophrenic, I call it. So part of it wants to stick to water, and part of it hates water. So as we heat it up, the part that hates water becomes more and more important in terms of determining whether it's soluble or not soluble. So that's why it switches. So it's not just this chemical, there's a lot of chemicals where you have one part that likes water, another part that hates water. They do this, and they all do it at different temperatures.